Biological membranes control the passing through of substances, so they essentially control what goes in and what goes out of a cell, but not only in and out of the cell itself, but even individual cell organelles. For example, if this is a mitochondrion with all the infoldings of the inner membrane, which I'm not going to continue drawing right now, substances have to pass a membrane to go in, and the membrane now controls what is able to go in and what is able to go out. So it's a semi-permeable or selectively permeable. And the point is that if membranes would allow all substances to go through, then why have a membrane in the first place? I mean, there's no point in having a membrane. These three components over here, they make up a membrane right now, and I'm going to explain these components right now and how they also influence or determine the passing of substances in and out of the through a membrane. So we really zoom in a lot now, and we really have a look now in very much detail at the actual construction of a membrane. And there are three parts: phospholipids, proteins, and cholesterol. Surprise, surprise. Many of you might think that cholesterol is bad for you. You should not eat too much cholesterol and so on. Well, I'm telling you right now, cholesterol is very important because there are highly important components of our cell membranes. Now, that phospholipids are our main component. The proteins can have different shapes, size, and form, and the cholesterol is also important for controlling the flexibility or fluidity of the membrane. And now let's have a look at how these three components play together. So that's the components of the biological membrane. And over here we have a more detailed look at a phospholipid. So over here you see again this schematic drawing from before, and you see that a phospholipid has a head part. We call this the head, hydrophilic head, and hydrophobic tails. And the hydrophilic heads are water soluble. So the water molecules arrange around the hydrophilic head without problems. And your hydrophobic tails are not water soluble, they're fatty. Phobia means being afraid of something, so it's afraid of water, and philic means loving. So this one is water loving and water fearing. And this is a single phospholipid. And over here you see a more realistic drawing or computer image of a phospholipid, and these little circles that you see over here, these are individual atoms. So the white spheres, these are all hydrogen atoms. In the middle over here you have a carbon chain, and the blue atom in the center over here, that's a phosphate, a phosphorus, and so on. So that is basically the a slightly more realistic drawing, and you can also see over here that there are these two tails of the phospholipid, and this is the hydrophilic head and the hydrophobic tails. Now if you put a whole bunch of phospholipids together and add some water to it, and if you shake the bottle a little bit, then the following is going to happen. The hydrophobic tails, the water-fearing tails, will arrange in such a way that they're together with each other. You see, so basically in the tails are together with the tails, and the heads of the phospholipids are turned outside towards the water. So there's water, H2 over here, and there is H2 over here. Now you might ask, why is there water? Well, we're made 70% of water, and if this is inside here is the cytoplasm, it's mostly water. So in the middle part over here we have the membrane is hydrophobic, of course, because this is where the tails are, and the heads over here and over here are hydrophilic. They're water loving, they're next to the water. And when a when the phospholipids arrange in such a way, we refer to the whole structure over here as a phospholipid bilayer. Bi means two layers, and this is one membrane. Some students sometimes get confused. They think that is basically that's two membranes, that's double membrane. It's not, because one membrane is made of a bilayer. And for some cell organelles have two membranes, well then you need two bilayers. So that's basically an overview of what phospholipids are, and let's move on to the next one. Over here, a term which I forgot to explain, amphipathic. What does this mean? A substance is amphipathic if it contains both hydrophilic and hydrophobic parts at the same time. So it is water soluble and not water soluble at the same time, or not at the same time, at different ends of the molecule. And substances that are able to do that are called amphipathic. I want to give you an example from real life. Soap, for example. In case you're wondering, soap is chemically similar to phospholipids because it's also amphipathic. This means on one side it is hydrophilic, it's able to dissolve in water, that's why soap and water mix quite well. On the other hand, soap is also able to remove fats and oils, because the other side of the soap molecule associates with the fats. So soap is very amphipathic, and if you really want to have a real life connection to phospholipids, think of soap bubbles. Soap bubbles being very thin, and you can also imagine that soap bubbles are actually very similar to membrane structures. So this was basically it concerning the phospholipids. Wow, I even explained it over here and forgot to show you. They have both hydrophilic and hydrophobic parts. And over here now, a more detailed look at a membrane structure. And what you see over here is you see the proteins embedded in the phospholipid bilayer, and over here the cholesterol. So you see all three parts here. There are a few more things that I'm going to explain, but I'm going to explain this in more detail in a different video.